Click for IB Biology. Here we're looking at the concept of gene knockouts, the introduction of deliberate mutations into genes so that we can analyze what those genes do in the phenotype. And we're also going to take a quick look at an application of CRISPR-Cas9 in resolving a genetic issue. There's a blank PDF of this presentation linked into the description below. You can download that and annotate with your own notes. Then you can also take the 10-question quiz at the end. The answers are there for you to check. And also I've provided an AI prompt that you can use in your favorite chatbot to set up a multiple choice individualized learning assessment. So let's look at the concept of gene knockout. So we start off with a piece of biology, which of course is that we have a gene. Normally we have a gene. And through the process of transcription and translation, which is protein synthesis, this is responsible for the characteristic of that organism. And what we observe as that characteristic is what we would call the phenotype. You might recall that word from study of genetics beforehand. So the gene is responsible, at least in part, for the production of the phenotype. So the argument is that if we knock out this gene, or if we knock out a gene, and then observe the organism, we look for what characteristic no longer appears. So we have a gene, we don't know what it does, and what we're going to do is we're going to knock it out, and then we will look at the phenotype of the organisms produced, and the characteristic which has changed or which no longer is present, that tells us what that gene is associated with normally. The knockout techniques include deliberately mutating targeted mutations of genes, often with chemicals. There's another technique which is now an old technique called zinc finger, ZFP. And what ZFP does is it causes this molecule called ZFP is introduced, and it causes the DNA molecule to be broken across the gene, and in that way preventing the formation of the natural phenotype. Today, the more common way of carrying out this is a much more specifically and precise targeting of the gene that we had up here. So we have a chromosome, we have a gene, and the CRISPR-Cas9 system can target that gene, break the gene deliberately with the enzyme carried as Cas9, and actually deregulate that gene, and then we know what it's actually associated with. So in the CRISPR-Cas9 system, there is an RNA, a guiding RNA molecule. That's what the G stands for, so guided. So the RNA is guiding to the gene the Cas9 enzyme, which is has the capacity of endonuclease activity. That is an enzyme which breaks down DNA or nucleic acids. So if we take a look at the successful use of a CRISPR therapy, we come back to sickle cell anemia again. And just a reminder that in sickle cell anemia, a point mutation, this is the normal pathway. Here we end up with transcribe and translate and add glutamic acid to the formation of the normal DNA. But with the mutation of T from A to T, there's the mutation there, the messenger RNA ends up adding valine, and the valine ends up producing this, these arrows are incorrect, ends up adding the protein which is known as the sickle protein, and that causes sickle cell shape, and of course that leads to sickle cell disease and the crises which can arise with that. So we have a technology that can correct this. Now, interestingly, the technique for CRISPR-Cas9 treatment of sickle does not attempt to correct the sickle cell allele itself. Instead, this relies on the fact that a person who is suffering from sickle cell, just like all of us, has genes. So adults have genes called the HBB gene. And what this does is produces fetal hemoglobin. So that's a fetal hemoglobin gene. Now, all adults have this, but it turns off as we reach the late stages of child development during the pregnancy. And we transition from fetal hemoglobin to normal hemoglobin. So we all have that, but this is suppressed, it's turned off late in child development by the BCL11A suppressor. So the BCL11A suppressor turns off the synthesis of the fetal protein. And what we're going to do is we're going to break this gene and we're going to turn back on fetal hemoglobin production. So the CRISPR-Cas9 has two parts, as I've already explained. There's the guiding RNA that takes it deliberately to this particular gene, and then the Cas9 enzyme splits the gene in the middle and disables it. The host organism, the host person, normally fixes the break, but that's already broken the actual BCL11A gene. So even though the suppressor is still broken. So in this way, what happens is that with the CRISPR-Cas9, we disable the suppressor, which turns back on the fetal hemoglobin. And so people with sickle cell condition are now able to generate fetal proteins, fetal hemoglobin proteins, to carry oxygen, and in this way they compensate for the loss of their normal hemoglobin. A very circuitous way of solving the problem, but very, very clever. There's a link to the channel here and just the playlist if you wish to go back over videos. We're now moving on to another part of the course.